Well, it's an absolute delight to be here. It's a fantastic meeting. I'm enjoying the scientific discussions, and it's also a beautiful city and the sun is shining. I'm also aware that I'm the last speaker of the day, and I've got a few things I'd like to tell you, so I will get cracking. So for many years, my group has been interested in the problem of cancer invasion, which is rather, I think, beautifully illustrated in this image here, but it's a somewhat tragic image because this turned out to be a fatal disease. But what you're looking at stained in brown here is a type of skin cancer, so it's a squamous cell carcinoma as opposed to a melanoma. This is the normal skin at the top, and it's invaded down here about 2 or 3 millimeters into the surrounding tissue. And you can see that it's invading, as a kind of collective tissue with some rather intricate architecture that you can see kind of here. Some areas seem to retain rather kind of thick strand geometry, which is something I will talk about in the first part of my talk, whereas other areas retain or have a rather kind of thin, almost kind of single file, form to their collective migration. The other thing that you can't actually see in this image, but I really need to tell you about, is that it's not just about the cancer cells. All this sort of white space here is full of extracellular matrix and other cell types. And actually, it's the combination of, the kind of genetic alterations and changes in the transcriptome of those cancer cells here in green, interacting with the extracellular matrix and all these other cell types in various different colors that really gives rise to the kind of phenotype or the behavior of the cancer. And we're particularly interested in spread, therapy response, and as I'll get on into the second part of my talk, actually the evolutionary dynamics in these cancer models. Because the different kind of colored nuclei in these green cells here are just a reminder to tell me, for me to tell you that actually not all the cancer cells within a tumor are equivalent, but they have some genetic changes, and part of the reason why the disease is so hard to treat is that you're effectively hitting a moving target. So if you have something that is good against, say these cells with the red nuclei, perhaps the ones here with the kind of purple nuclei don't care, and they then become dominant, which is one of the reasons why the disease is really challenging to treat, and actually one of the things that we would love to be able to actually make some headway in terms of tackling. So this is, how we got into this. We started, actually taking explants of patient material, like that tumor that I just showed you, putting them in kind of 3D culture, and then observing what happens. And we saw a very kind of close interplay between the cancer cells in green here and one of those other cell types that I showed you on that previous slide, which was the cancer-associated fibroblast. So these cells here in pink. And actually, you don't need to worry about any of the other cell types in this cartoon for the remainder of this talk. We could kind of demonstrate that the fibroblasts were functionally very important, because if we made a spheroid of cancer cells alone, as is here, it's not very invasive. If we do this with cancer-associated fibroblasts added in, it's much, much more invasive. And we've unpicked a range of different mechanisms by which fibroblasts promote the invasion of cancer cells, some as the result of actually very stimulating collaborations with Javi Trepat. But for the next kind of 5 or 10 minutes, I'm not actually going to talk about the fibroblasts. I'm just going to talk about what determines the kind of geometry of these strands that you can see here. Are they thick strands sort of broadly pushing, or are they kind of more narrow strands kind of infiltrating between spaces? So this is effectively just a re-articulation of the problem now. Another example of a squamous cell carcinoma stained actually in a slightly different way. But the other kind of point to make is that it initially starts off somewhere up here, and then it's almost like a layer kind of almost invading down. You can imagine that there might be some sort of self-generated gradient kind of acting in this direction, but then when it sort of penetrates a certain distance, actually it's a little bit more kind of encapsulated and a bit more like a spheroid, perhaps with a slightly different type of self-generated gradient that one might imagine being a little bit more radial in its kind of architecture. The other thing to kind of highlight is that actually there are a range of different ways in which we can measure this type of strand geometry. One of the ways that you'll see in a couple of slides is that we just measure the number of neighbors that a cell has. So something like this, will have more neighbors than a cancer cell in a strand like this, which is, I think, fairly intuitive. So, how do we study this? Again, we do some sort of explant cultures, but we also use kind of reductionist kind of co-culture models and computational modeling, as I will tell you. So these are actually kind of four different fields of view of the same tumor explant, which we've now just put in one of those collagen-rich ECMs that you've heard about from previous speakers. You can see some single-cell migration. There's actually a bleb-driven sort of migration in this cell here that then switches to a lamellipod type of migration. happy to discuss that later. But you can also see, again, these different kind of strand geometries here. Single file, kind of 2 or 3 wide, and then, this kind of broad pushing front here. And the fact that, even in a homogeneous kind of collagen-rich extracellular matrix, we're getting these different types of behavior suggests to us that actually there might be some variation between, the tumor cells in these different regions that is giving rise to these different behaviors. So what might those sort of, variations be? So, our aim, in this first part is to really understand, what determines the collective organization of these invading groups of cancer cells. And there are a range of possibilities. Some are up on the screen. I'm sure you might be able to imagine more of them. The challenge for us, and particularly me as somebody with an experimental background, is that when I have more than about 2 parameters to vary, I get, really kind of stuck in terms of trying to unpick which might be having the dominant effect over the other. So this is where, actually collaborating with, theorists and people who are able to build computational models has really proved, very stimulating and I think invaluable for the research we've done over the last few years. So you've heard about this sort of modelling previously. It's another version of a POTS model with many of the same features that were kind of outlined earlier. The only, I think, sort of a couple of significant differences. One is actually that the cells have an ability to remodel their surroundings. They can degrade the extracellular matrix. The other is that over time, there's actually growth in this model, so they can accumulate biomass. If they accumulate sufficient biomass, they divide into a couple of cells. I won't kind of spend more time discussing the ins and outs, but happy to answer questions on it. This is just to show you that actually, we can generate, simulations that look a little bit like the experiments I've shown you. If we don't have any fibroblast, you just get a little bit of growth over, a period of computational time. If we add fibroblasts, they're kind of in similar kind of positions, leading the invasion of those strands in the spheroid model, and you get this invasion down into the underlying kind of in silico extracellular matrix if we have a layer experiment. And this is just the kind of some of the experiments that we use to parameterize the model. So with this in hand, we can actually vary all of those parameters kind of in multi-dimensional space and then ask, which of those sort of parameter spaces give rise to different patterns and different extents of invasion. So this is just to kind of graphically show that we can get different forms of invasion. This is a sort of broader, kind of more pushing front. This is a thinner strands here, and we can get similar variations in those sort of spheroid models as well. We can then actually measure a range of different parameters, including neighbor number, the extent of invasion, and whether there's growth or fracturing of the objects, and then kind of plot all the kind of result of all the simulations in these sort of PCA plots. So this is rather busy and complex. I'll just very quickly tell you about a couple of things that we've learned from this. This is exactly the same PCA plots, but actually all that's happened now is we've just shaded them based, in this light blue color on the value of the extracellular matrix proteolysis term. And you can see actually that there's a good relationship between the proteolysis term and the number of neighbors, suggesting that high proteolysis might be related to wide strands. It's also related to population growth, something that I will come back to. Although intriguingly, in neither the organotypic nor the spheroid context did we see much of a relationship between extracellular matrix proteolysis and the maximum invasion depth, which the cells were predicted to achieve, suggesting it's not really that proteolysis determines how far the cells get, it's that proteolysis determines the kind of, the pattern with which they're invading. So that's, a prediction. Is it true? So what we did was we played around with this model and we either, turned up or turned off their ability to degrade the collagen-rich extracellular matrix. And what I hope you can appreciate here is that when we overexpress the relevant collagen protease here, we get much wider strands, and when we knock it down, the strands are still getting almost the same distance, kind of down, but they're much, much thinner. And we see similar things in the spheroid context here. So really telling us that the predominant thing that proteolysis is doing is, determining the breadth of the invasive strand, not how far it's getting. If we think about, the relationship between cell-cell adhesion and, the invasive pattern, we can now see, a slightly different thing. There seems to be somewhat of a relationship between the maximum invasion depth, particularly in the organotypic model, and also a little bit of a relationship to population growth. Again, we can explore this experimentally by playing around now with cell-cell junctions, either depleting alpha-catenin from the system or knocking out both E and P cadherin. And one of the things I'd like to sort of draw attention to here was this, I think, somewhat unexpected result from the model was that actually in this kind of layer-like situation here, where we think there's a uniform kind of queue that's making the cells invade down, that we see a positive relationship between cell-cell adhesion and the extent of invasion, something that we could confirm experimentally as shown here and quantified here. However, in the spheroid context, there's a much sort of flatter relationship. And again, this was something that we could confirm experimentally. You can hopefully just see that you get the same sort of extent of invasion out here as you do when we've knocked down alpha-catenin here, again quantified. So this suggests to us actually that cell-cell adhesion per se is not important for the cells to be able to invade, but there's some sort of context-dependent role for it. And what might that context-dependent role be? And there are a couple of things that are different between these two experimental contexts. The first is that the organotypic assay both has this layer kind of construct, and we impose on the model a uniform kind of chemotactic queue, whereas here the cells are initiated as a circle and with a radial kind of chemotactic queue. So we've got two things that are changing at once, which means we can't really determine what the difference is between this, why we get this relationship and why we get this relationship. But obviously it's a model, so we can play around with it in ways that are very difficult to achieve experimentally, and we can make it initiate as a spheroid but with a uniform gradient, and now we get a very kind of clear kind of indication that there's relationship, positive relationship between cell-cell adhesion and the extent of invasion kind of comes back. Suggesting that perhaps what the cell-cell junctions are doing is actually promoting coordination between cells rather than just adhesion. And to cut, I think a very long story short, what we think is going on is actually that, cell-cell adhesion in this context is regulating the formation of this supracellular actomyosin cable, perhaps a little bit analogous to these ideas around the importance of, cell-cell junctions in regulating, surface tension and cell sorting and cell migratory dynamics. And that actually this is lost when you knock out alpha-catenin. And we've employed an orthogonal method to interfere with this kind of supracellular actomyosin kind of cable by kind of individually activating, Rho kinase or ROC in all the cells, which messes up, the supracellular cable, giving you a situation like this. And this also narrows the strand widths, suggesting that there's this relationship between cell-cell adhesion and the patterning is because cell-cell adhesion is actually important for regulating the supracellular actomyosin coordination of those cells. I think I've already told you this. Lastly, for this kind of first half of the talk, what we've also been able to observe and actually challenge the model is that even though it was done in a relatively simple context, we wanted to see if any of this applied in vivo, because what it was telling us was that actually cell-cell adhesion was important for invasive capability, particularly that initial invasion down into the surrounding layers, and that matrix proteolysis seemed to be playing an important role in determining strand width, but also in determining that growth parameter. So actually there seems to be a very good correlation between the strand width and the ability of the cells to form lymph node metastases. And this, I think, is rather interesting because it suggests to us that actually the coordinated behavior of these epithelial malignancies is really important for their metastatic behavior, actually putting a slightly contrary point to the idea that it's all about epithelial to mesenchymal transitions in terms of cancer metastasis. So just to kind of summarize this first part, I hope I've been able to kind of convince you that multiple different kind of collective invasion patterns can be present in the same tumor. We don't think that there are particular distinct categories, it's more of a continuum. These are regulated both by matrix proteolysis and cell-cell adhesion, less so by cell matrix adhesion. I haven't had time to show you that data. We think that cell-cell adhesion is enabling supracellular coordination, potentially analogous to surface tension. And just finally, that track widening and tumor growth are linked features when there are insufficient CAFs to generate space. And we think this is part of the reason why this kind of track widening behavior is very positively correlated with lymph node metastasis. So now I'm going to return to this image but switch gears for the second half of my talk, which is now kind of thinking over much longer kind of both time and length scales, because those invasion processes are things that happen over maybe 2, 3, 4 days, perhaps a week or two. But cancer evolution actually plays out over much longer time scales, months or in some cases years. And actually what we need to kind of think about is actually not that all these kind of cells here in brown are the same, but actually they have some variability in both their genetics and their phenotype, and actually they're somehow kind of competing in response to the challenge of both surviving and migrating within this environment. But actually, when one thinks about this, this is rather a large kind of length scale here, almost a centimetre or so. So, to what extent are these cells really in competition with these cells? And actually, perhaps this is, in some way determined by actually how migratory these whole kind of collective of cells are and how much they're interchanging positions. So is it a mixed or a well-mixed population? And so in the last kind of 10 minutes or so, I'm going to tell you about some work that's kind of shifted scales a little bit, shifted gears, where we've built a different type of model, in this case, an agent-based model where each agent represents a single cell, and we're looking at the kind of larger, longer scale colony dynamics. It's a relatively straightforward model. It has repulsive forces so that cells can't occupy the same space, adhesive forces because we're modeling sort of epithelial cancers here, and kind of crucial to the model's behavior is we have this sort of rather, well, perhaps obvious assumption that if the cells don't have enough space, they're not able to proliferate. And this is really kind of key for the emergent behaviors, as you'll see. The reason why we have that assumption is because in many, many examples, now in a lung cancer model, we see that the denser the cells are, the less proliferative they are, as measured by how many of them are in S phase that you can see here. So what do we get when we do this? This is now the model simulation. The magenta kind of dots are meant to be cells that are proliferating. So what you can see is that over time, and this is meant to simulate, I think a couple of weeks or so of, cell growth, that you get this sort of kind of tumoroid or whatever with a non-proliferative center and actually a proliferative margin. And so now what we can sort of start to hypothesize is actually that if we have kind of ongoing mutational processes in this sort of a model, it's actually going to make a difference whether a mutation is born here or whether a mutation is born here. Because if it's born in a non-proliferative zone, in a sense it's going to have to work harder to actually become dominant and take over the tumour. Whereas if it's born here, it's a somewhat advantageous position because it's likely to be proliferating anyway. So how can we kind of study this? Well, one way that we can study this is just by actually generating these sort of cancer models, but by switching the colour of cells at defined points. And this is the sort of technology that we use to do it. I won't go through the details, but effectively what it means is that at a defined point in time, we can label a fraction of the cells different colours. So now we can run the simulation again, and I hope it will play. Yep, here we go. And here we do the colour labelling. 3 different colours because that's how we do it computationally. And what this actually leads to is this emergent kind of formation of spots and streaks. So these are the cells that were born at a favourable place, and even though there's no advantage to these cells, they tend to form these kind of colonies or subclones that do rather well, whereas if the cells happen to be born in this disadvantageous place, actually they don't do very well. So that's a model prediction. Is it true? Turns out that it is true. So now what you can see, this is just after labelling, and you'll see the movie play for about another week. And you can see that these cells in the middle kind of get stuck. It's a rather kind of bleached out movie. But the ones that were kind of near the edge form these streaks, and you can also see them here. So what's happening here is that the emergent kind of property of this model is that in the middle it's getting kind of jammed. Actually, this relates, I think, to quite well to a lot of the cell jamming literature, but you're getting this sort of almost ballistic type migration of these cells outwards round the outside. So that's all well and good. We can kind of create a model that replicates our experiments. But what about playing around with some of the conditions? Because not all tumours look like this. This is rather oversimplistic. And despite its simplicity, we were kind of gratified that when we looked in vivo, this is now some of these tumours growing in a lung, we could again see this kind of divergent behaviour with these ones, these subclones that were labelled near the kind of edge, actually kind of proliferating outwards, whereas these little ones that kind of were closer and up against the chest wall didn't proliferate very well. So this is really telling us that both in this in vivo context and in our simple model, we have actually this place of birth effect being rather strong. And we can measure it just simply as the variance of the subclone size. So a big variance means a big place of birth effect. A small variance, which if we do the labelling rather late, doesn't have time to develop, means a low kind of place of birth effect. So this is what I've just told you. Cells in the interior do not proliferate due to the compressive or the build-up of compressive forces. But is it really as simple as this? And one of the great things, and one of the things I really love about the kind of computational models, is that we can play around with parameter space and really see what might be possible if we play around with the various kind of parts of the model. So what we've done here is we've kind of reduced that viscous drag term, and again you'll see these sort of simulations. But because we've reduced this viscous drag term, actually the cells really don't kind of get quite as stuck in the middle. They still kind of continue to move past each other, and this leads to a slight disruption of this spots and streaks pattern. So how might we experimentally, achieve the same thing to ask if this really is possible and this could happen? We've used TGF-beta, which is a well-known way of driving, the kind of intraepithelial migratory dynamics. This is just to show that it does exactly that. This is sort of PIV kind of tracking, again, in a kind of a layer assay. But just the key point is that TGF-beta drives, the migratory dynamics of these epithelial tumours. Also, if we now do the labeling, what you can see is that TGF-beta reduces actually this kind of spot and streak pattern that you can see here. It leads to more proliferation in the interior, and actually this kind of place of birth effect as measured by the standard deviation of the subclone size is dramatically reduced if we treat these colonies with, or these tumour colonies with TGF-beta. And this can be partially reversed if we block the migratory dynamics by interfering with the function of the formin moderators of actin polymerization. So into the last sort of 5 or so minutes, what I'd like to sort of kind of take you on a quick tour through is trying to understand if any of this is relevant outside of our experimental models of, in this case, lung cancer. So, coming back to human disease, this is another image, of a sadly lethal tumour. But what we can't do in the patients is we can't do all that kind of Cre recombinase thing of labeling the cells with different fluorophores, but we do have lots of sequencing. And actually, we've been collaborating with the group of Charlie Swanton, who has done a lot of sequencing of very, very many lung tumours. And this is actually what we've been able to leverage to infer whether these tumours have high extent of collective migration in vivo or not. So how do we do that? Again, we turn to the modelling, and what we do in the modelling now is we don't try to simulate, those kind of spots and streaks patterns. We try to simulate mutational processes that are ongoing in that model over time, and we, each of those mutations has an identity, so we can then do a virtual sequencing experiment of those in silico tumours, and from those we can generate these sort of patterns of sequencing data which are widely available for human disease. Basically saying, if you have a mutation, is it present just in a few cells, or is it present in almost all the cells? Because these tumours are mutating at an ongoing rate, you see that you have many, many mutations that are just in a subpopulation of cells because they arose relatively recently. And occasionally you see a sort of bulge here, which is indicative of an actively expanding clone. So we can use our model to predict these what are called variant allele frequency distributions. This is just showing how we can run the model now either with some selective advantage or disadvantage to the mutations that are induced. Actually, this doesn't dramatically disrupt the spots or streaks pattern as you might expect with negative selection. The streaks tend to terminate. So we can then do this, we can do the in silico sequencing. I won't belabor the point, but what we're able to do is basically identify if we're getting place of birth effects, because they show up as little bumps in these kind of plots here in this region here. Happy to discuss that more if people are interested in that at the end. So we can then take our in silico data and model what a tumour with high levels of mixing would look like if you sequenced it, or a tumour with low levels of mixing would look like. So we can then use our model data. It's kind of come up a little bit bleached, but we're kind of plotting it, some of the metrics we can generate from the sequencing data in silico in 3D space. And actually, rather gratifyingly, real, human lung cancers sit in a very sort of similar space. And then from this, we can infer whether they might be mixed to a high degree or mixed to a low degree. So with this information, we can then correlate that with other aspects of the disease, in particular their transcriptome. So this data I was incredibly gratified to see, because it tells us that the tumours that tend to be mixed, so these ones at the orange end here, have high levels of TGF-beta signalling and high levels of this epithelial to mesenchymal transcription factor here called ZEB1, really supporting some of the ideas that were coming from that TGF-beta treatment in vitro. And those, as I should remind you, came from ideas from the modelling. So the kind of last couple of things that we noticed when we were going through this data is that a lot of this sort of mixed signature that was associated with TGF-beta and EMT turned out to be driven by markers of one of those cell types that I've already showed you from the tumour microenvironment, which are the cancer-associated fibroblasts. So here, actually, you can see many of these CAF markers are high in the mixed genes and low in the unmixed genes. So we then kind of thought, well, perhaps, this could be telling us that actually, fibroblasts we know drive migration in this system. Perhaps fibroblasts are driving this kind of mixing of the tumours, and they're driving this sort of signature that we're seeing. So we went back to our experimental systems, now doing kind of co-cultures of these models with the cancer-associated fibroblasts, two different ones called CAF 796 and 845 here. And what I hope you can see is that this sort of spotty pattern in the middle, the spots and streaks pattern of the controls with the spots in the middle is somewhat lost when we treat or co-culture with the cancer-associated fibroblasts. So we get these central growing subclones, and they're very much intermixed with their neighbours, really telling us that cancer-associated fibroblasts can drive mixing and reduce the place of birth effect. And this very much correlates with migration. Two more quick slides to show you, to really kind of try to wrap this up and relate how we think that this might actually relate to the really lethal aspect of cancer, which is not just its propensity to metastasize, but actually, how it relates or how it responds when you treat it with a therapy and how it's able to evolve. So now what we're doing with the modeling is we're treating it with an in silico therapy. You see it sort of regresses for a bit and then comes back, and it comes back in these slightly darker colours. The slightly darker colours indicate that there's been selection for a gene that is associated in silico with being able to resist killing by the virtual therapy that's applied. So now we can ask, is this kind of mixing behaviour and the ability of the cells to interchange position within the tumour actually affecting the rate at which those resistant clones become dominant? So we're not changing in any way the mutation rate in these models, we're just changing the mixing rate. And what you can see in the red lines, which represent the mixed tumours, is that mixing, particularly if we have higher drug concentrations, really favours the rate at which those resistant clones can become dominant. So actually, a well-mixed population in these models tends to evolve to a fully resistant state faster than a population that is not well-mixed, which is something we're really excited about thinking for the future. And just last bit of data here, we can actually now model this by making one of the colours, in this case the yellow colour, associated with the drug resistance in vitro. And you can see that if we have a control here, we get equal red and yellow. If we treat with the drug, we skew towards getting more yellow over red. This is the control situation where we haven't done any mixing. If we drive mixing, now the yellow is able to dominate much quicker or much more effectively than if there's no mixing, at least proving an in vitro proof of concept to this idea that high levels of mixing and cell migration within tumours is really driving faster evolution. So these are my concluding thoughts. The ones in grey I've already, I think, highlighted, but I'd really like to leave you with this message that, perhaps, the bad subclones are able to dominate mixed tumours faster, and that this provides a sort of, a tumour-level mechanism linking epithelial to mesenchymal transition and cancer-associated fibroblasts to therapy resistance. So I realise I've spoken for a couple of minutes too long, but it would be remiss of me to not really acknowledge all the people who've done the work. The first part was led by Takuya Kato and Rob Jenkins, and the second half was done by AJ Bhargav, Xiao Fu, and Sasha Bailey. Thanks to funders, thank you for listening, and I'm happy to answer questions if there are any.